So we'll talk about the unknown first. So this is an example of kind of what your unknown will look like, except that you'll have pictures of things, right? Since we cannot actually do any lab work. But this is the same kind of handout that you would do in lab. It's just that you would do the actual work, you would make drawings, and you would interpret what you see. So here we're giving you images of the different things. So each of you will get your own unknown sheet. It'll have an unknown number here, and you put your name in, and then there'll be a picture, consistency, is it dry or mucoid? Is there any pigment present? There are different ways you can edit this document. You can underline, you could circle things, or you could highlight, like here, I highlighted in yellow. So you just need to be able to let me know what your choice is on the handout. So, pigment and consistency, and then an image of a gram stain, is it positive or negative? An image of a spore stain, positive or negative? Image of an acid-fast stain, positive or negative? Do you have a rhodcoccus or spiral? And the predominant arrangement. And then, based on all of your data, you need to identify it. So you need to give me a logical organism, right? The genus and the species that matches all of these data. So it must be a logical guess, right, based on all these characteristics. And you cannot use any uncommon, or strange bacteria or any dangerous pathogens, because it has to be something that we would give you in lab. So it is useful to go back and look at all the handouts and see what organisms we've talked about. But, this is all you have to do, is fill this out and then, give me a logical identification for what it could be. And there can be more than one, right answer for these. Although for some of them, there really is just one, but for some others, more than one answer will be acceptable. You have until tomorrow night to turn this back into me, because normally it would be turned in at the end of the lab period. So you would do all your work, make all your smears, examine them in the microscope, make observations of the growth, and make a guess as to what it was, all in a 2-hour lab period. So I'm going to give you till tomorrow night. So that's a lot of time, and this is much easier than the way you would normally have to do it. So any questions?
So the pictures are all the same organism?
Yep.
Okay.
All the pictures are just for one organism.
Okay, got it.
Now, when you're ready to submit, I can't see, yes, after class, after we're finished, I'll send them out in an email. Oops. So it's important that you do submit these. Yeah, I can't see chats when I'm sharing. So you have to submit this through the assignment tool, right? So make sure you submit it. Remember, no Apple formats. If you save this document and it saves it as a .pages, I will not be able to open it. Blackboard will not be able to open it. So you have to use a PC compatible format, either .doc or .docx or a PDF, but no Apple formats. And so make sure you submit this by tomorrow night. No, you don't have to print it out. It'll be emailed to you, so you can edit the document and then just save it and then send it back. Any other questions about the unknown? Obviously, you can't see the lab exam yet because it's hidden from you. But on Wednesday, your lab exam will start at 1 p.m. You have an hour and 15 minutes to complete it, so you have until 2:15. At 2:15, the exam will auto-submit whether you finished or not. A calculator is on the home page, but it's not a scientific calculator, so if you need to do anything, logs or exponents, you can use your own calculator, but there's really nothing on here that requires a calculator, just your brain. But before the exam begins, you must go to Blackboard Collaborate and join the Lab Exam 1 session that's there and turn on your camera so I can see you. And then you can go to the Week 7 course content and open up the exam. But you got to turn on your camera. If your computer doesn't have a camera, then use your smartphone or tablet because you can dial in to the Collaborate session on your phone. So I have to do this because, as I may have mentioned, I caught 2 students cheating on the first exam. So now I have to make changes. And this seems to be the easiest way to do it. I don't remember if it was someone here that was telling me about ProctorU. Is anyone familiar with ProctorU? Are you using that in any of your other classes? I can't say, is that a yes or a no?
No.
No? Well, ProctorU is very strict. I mean, they watch you like a hawk. If you have any unusual movements, you are shut down from taking the exam and a report is sent to your instructor. So if you happen to suddenly look, to the left or the right, or they catch you staring down in your lap, the exam is over for you. So they're very, very strict. I, don't want to use ProctorU, right? And the other alternative was something called a lockdown browser, where once you open the exam, that's it. You're stuck on that page. You can't go anywhere else on your computer, right? You're locked down. But the problem with that is if you get disconnected while taking the exam, you're locked out. And then you got to contact me, and I got to go and change it for you so you can sign up again. So, this seems to be sort of the easiest way to do it and show that I'm making an extra effort to deal with cheating. So, any questions about how that's going to work?
For the plate count, could we just use a paper, like a scrap piece of paper?
Yeah, and then just after you're finished, hold up the scrap paper so I can see it.
Okay. So, and then if we have any questions during the test, we could just send you a chat. We don't have to, we shouldn't email you, right?
Right, just send me a chat because I'll be there watching.
Okay. And then, so that means we would have 2 tabs open, the video and the test?
Well, no, you will just have you'll just be looking at the exam, right? Your camera will be on, but you won't see that. And I'll just be watching all of you on the screen. So all the little boxes with all your, smiling, happy faces, I'll be seeing all of them while you're taking the exam.
Right. So in another, like, we wouldn't be seeing you, we would just be seeing the exam page while this tab was open, basically.
I'll just be seeing you. You will be seeing just the exam.
Right. Okay. Got it. Just making sure.
Yep. So any other questions about the exam?
So for the lab, we can't use our lab handouts either.
No. It's an exam, right?
Okay.
Not allowed to look at any other materials. But for the unknown, you can look at anything, right? For the unknown, but not the exam.
Okay.
Right? The exam is an exam,
Mm-hmm.
right? Not supposed to look at anything else during an exam. So any other questions? Okay, so let's do some review then. I'm just looking at the syllabus so I can keep track of what I'm talking about. So we did start talking about lab regulations and lab safety. Even though we're not physically meeting in lab, you still need to understand, lab safety regulations. And so, of course, there's something that you're always supposed to do before you start lab and before you leave the lab. There's actually two things you're supposed to do before you start and leave the lab. What are those two things? And talk to me, no chat.
Is it disinfect the table?
You have to submit the tabletops, and what else? What else do you do?
Wash your hands?
Yep, wash your hands. So right before you start lab, you disinfect the tabletops, wash your hands. And before you leave the lab, you disinfect the tabletops and wash your hands, right? So, things that we normally would have done, but we're not doing because we're not meeting. But you need to understand, those general practices. We went through the lab basics handout where we talked about different tools and equipment used by microbiologists. And we did talk about pipetting, which we'll get to in a couple of minutes, and also about aseptic technique, which we'll get to in a minute, using loops and things like that. For the environmental plate, where do we find bacteria and other microorganisms?
Like, what were all the places that we saw?
Or just where do you find bacteria and other microorganisms? One word.
Everywhere.
Everywhere, right? They're everywhere. So then where can contamination come from?
Anywhere.
Right. Everywhere, right? So that's something you always have to keep in mind as you are working in the lab. The contamination can come from anywhere, so you got to be really careful, right? And when you were looking at the plates, at the different environmental plate results, could you tell that there was more than one kind of microorganism growing on those plates?
Yes.
Yes, and so how could you tell?
From like the shape, the color, size.
Right, so all the different colonies were not exactly alike, right? They had different sizes, shapes, colors, textures. And so you could tell there's more than one kind of microorganism present on the environmental plates. And then we talked about microscopy. So you should be familiar with all the parts of the microscope and what they do. You should be familiar with how to calculate magnification, right? Ocular times objective. The ocular lens magnifies 10 times. And we had 4 objectives, 4, 10, 40, and 100, right? For a total of 40, 100, 400, and 1,000, right? Then we started learning aseptic techniques. So how to flame a loop, how to hold tubes. So when it comes to tubes, is it okay to lay a tube down on the table?
No.
No. So that tube is either in your hand or it's in the rack, right? You never lay a tube down on the tabletop. So after you flame your loop, you make it glowing red hot, right? And so is it a good idea to immediately go get your sample as a way to minimize contamination? Or should you wait and let the loop cool down? Hot loop or cool loop?
I think you should let it cool down for, I
Right,
think we said 10 seconds or something.
right. Yeah, so let it cool down for like 10 seconds. And so you're not going to open a tube or a plate until you're ready to go in. So after you flame the loop, after it cools, then you can open your tube or plate to get your sample, right? But not before. And then always flame your loop when you're finished with everything, right? To make sure you kill whatever might still be on it. So always flame the loop before you use it and flame it after you use it. Then we talked about making smears, right? So you get a slide, you put a drop of water on, you get a loop full of bacteria. Remember, you touch and you look. If you see something, stop touching. If you don't see anything, touch again, but you want to stop as soon as you see something because that's more than enough bacteria. And then you flame the loop, cool it, spread the bacteria throughout the water drop, and then you dry it on the hot plate. And so what should your smear look like when it's dry?
You should barely be able to see
Right,
with
so
your eyes. Should it be thick?
right, very, very slightly cloudy. You should be able to see something, but not easily. You don't want it thick, you don't want it dark, you don't want it clumpy, right? So a nice thin, even smear. And then what you have to do with that smear before you start staining?
Heat fix it.
Heat fix it, right? Always heat fix, right? By passing the slide through the flame, 3 times, 1, 2, 3, and it's ready. And then we talked about simple staining first, so just using one dye, right? We give the bacteria color, and that enables us to see 2 things. What 2 things can we see? When you give the bacteria color, what 2 things can you see?
Shape and arrangement.
Right, shape and arrangement. And so what were the 3 common shapes?
Cocci, bacilli, and spirals.
Right, so rod, coccus, spirals. And the 3 common arrangements are clusters, chains, and single, right? But we did talk about one special cluster. What was that special cluster?
Tetrads.
Right, tetrads. And what organism had tetrads?
Micrococcus luteus.
Yep, Micrococcus luteus. But shape and arrangement, that's not enough information, right? What do we really want to know about those bacteria?
If it was gram positive or negative.
Right, because that tells us what kind of cell wall the bacteria have, right? So we always do a gram stain first, because we want to know, is it a gram positive cell or a gram negative cell? And knowing the cell wall is important because it's the first step to identifying an unknown organism. It can indicate the best antibiotic to use. It can also indicate the possible source of contamination, where the organism came from. So you always do a gram stain first. All right, so we want to know what kind of cell wall. So how do we do the gram stain? What are the 4 components?
First you put the crystal violet.
Right, the primary stain is crystal violet.
Then
Then we
you
rinse that off and then add
the iodine.
iodine, and then
And
rinse
then
that off.
decolorize. And then
But decolorize with what?
alcohol.
95% ethyl alcohol, right? And then we counterstain with what?
Safranin.
Safranin, right? So by the time we're finished, what's the color of gram-positive bacteria?
Purple. Purple.
Yeah, purple, right? So just remember, positive purple, right? PP. And the negative cells are red. So you should be familiar with examples of those bacteria. So if I asked you for an example of a gram-positive coccus, give me the names of some organisms. Like what?
Micrococcus luteus.
Right, Micrococcus luteus. What else?
Staphylococcus aureus.
Yep, Staphylococcus aureus, Enterococcus faecalis. What about some gram-positive rods?
Bacillus cereus.
All right, so something like Bacillus cereus or Bacillus subtilis. Gram-negative rods?
E. coli.
Right, like E. coli or Serratia marcescens or Klebsiella pneumoniae. Right, so you should be able to give me some examples. Not spell them, because remember the lab exam's all multiple choice. For the acid-fast stain, what genus do we use the acid-fast? Which one?
Mycobacterium.
Right, Mycobacterium. Remember that thick waxy cell wall? And so we have to use heat in order to help the stain penetrate. And so what's the primary stain for the acid-fast stain?
Carbonyl fusion.
Right, carbonyl fusion. So carbonyl fusion, and you put that on the smear for 5 minutes and then rinse it off. And how do you decolorize?
Acid alcohol.
Right, acid alcohol, and then counterstain with
Methylene blue.
methylene blue. So acid-fast positive bacteria should have that red fuchsia color, the purplish red or reddish purple, or some call it hot pink color. And then everything else should be blue, right? Because a mycobacterium is the only thing we're worried about that should turn that fuchsia color. Everything else should be blue. So if you did a sputum smear and you saw those fuchsia rods, what do you have?
TB.
TB, right? So you never want to see anything that's acid-fast positive in a sputum smear. Now the spore stain is another stain that requires heat, right? Because heat is needed to penetrate those spores. And so what's the primary stain for a spore stain?
Malachite green.
Malachite green. You leave that on for 5 minutes, and then how do you decolorize?
With water.
With water. Just rinse off the slide really well with water, and then counterstain with
Safranin.
safranin, right? So that way the vegetative cells will be red and the spores will be green or kind of bluish green in color. And what are the two genera of spore-forming bacteria? Which two groups make spores?
Bacillus and Clostridium.
Right, Bacillus and Clostridium. And what's the purpose of spores?
Or survival.
Survival, right? Then we talked about negative staining, which is something that we do not do in lab. We don't actually do any negative staining, but we just talk about it. And the best example was a capsule stain, right? That could have a red or pink slide, which had clear areas around the cells. And so remember, with a negative stain, whatever you are trying to see is not stained. Everything else is, which is why you can see that which is not stained. So with the capsule, the glass slide is red, the bacteria are red, but there's a clear area around the bacteria, and that's the capsule because it does not stain. What is the function of a capsule? What does a capsule do for bacteria?
Protection.
Right, protection from phagocytosis, right, from the immune system. So the thick layer of polysaccharide is protective. It's also sticky. We also mentioned the nigrosine stain, a black dye, which sticks to the glass slide, but not to the bacteria. So they show up as clear, cells against a black background. The main idea is that whatever you are trying to see is not stained, but everything else is, which allows you to see what is not stained. Then we talked about motility. So if bacteria are motile, what structures do they use for movement?
Flagellum.
Right, only flagella, right? No cilia, no pseudopods, just flagella. And if they have flagella, they occur in 4 different arrangements. What do you call it when they're covered with flagella?
Peritrichus.
Peritrichus. If they have a cluster of flagella,
Lophotrichus.
yep, and one on each end,
Amphitrichus.
and just one,
Monotrichus.
monotrichus, right? So you should be able to recognize the arrangement when you see it in a slide. We also use motility agar and look to see if the bacteria migrated from the stab. And there was something in the agar that helped us to see them. What was in the agar? Remember it turned red?
TTC.
Right, the TTC, the tetrazoium chloride. It was just there to help us see where the bacteria were. Right, so if they spread out from the stab, they're motile. If they're still in the stab, they're non-motile, right? So for micropipetters, the idea is to be able to know what they're set for. Remember, they have 3 numbers. So if this were the 0.5 to 10 microliter pipetter, how much would that be? If this was a setting on the 0.5 to 10, yeah, you got to talk to me. How much is that? So somebody say it.
Are you able to repeat the question?
So if this is the 0.5 to 10 microliter pipetter, what is it set for? What volume?
Is it 10 microliters?
Yep. If this was the 10 to 100 microliter pipetter, what's it set for?
100 microliters?
Yep. And if this were the 100 to 1000 microliter pipetter, what's it set for?
1,000?
1000, right? So again, if it's the 0.5 to 10 microliter pipetter, what is it set for?
.5 microliters?
No, 0.5 would be 0.005.
Is it 5?
Right, 5 microliters. If this is the 10 to 100 microliter pipetter, what's it set for?
50.
50. And the 100 to 1000 microliter pipetter?
5.
100.
500, right?
Yeah.
500. So you need to know what the settings mean on the pipetter, right? So for bacterial growth and the standard plate count, right? Serial dilutions. Remember that your dilution is the part, right, divided by the whole. So it's the volume added over the total volume. So if we added 1 microliter into 9, that's a 1 to 10 dilution, right? And that was 0.1 or 10 to the negative 1. If we added 1 microliter into 99, that would be 1 over 100, or 0.01, or 10 to the negative 2. But most of the time we just use 9 ml a tube, so they're all 1 to 10 dilutions. And remember, they are cumulative, right? So if we first did a 10 to the negative 1, and we did another 10 to the negative 1, and another, and another, what's the cumulative dilution at this step?
10 to the negative 2?
Right, 10 to the negative 2. And here?
10 to the negative 3?
Right, and 10 to the negative 4, right? So your dilutions are cumulative as you go. But you remember the final number that we need is not the dilution, it's the dilution factor, right? And what's the dilution factor?
Is it 1 over the 10 to the negative exponent?
1 over the dilution, so 1 over a negative exponent, and just becomes a positive exponent, right? Now, this only works exactly like this if you transfer 1 milliliter of sample onto the agar plate, right? Because you always have to multiply the volume added to the plate times the dilution in the tube. So if it's 1, then nothing changes, right? Because you're multiplying by 1. But what if you were multiplying by, or you were adding 0.1 milliliters? So you have to factor this into your calculation, right? And so you can do it at this step here, right? Because then you would have, 10 to the negative 3, and 10 to the negative 4, and 10 to the negative 5 if you used 0.1 milliliters. And so that would change your dilution factors, right? If you're using something other than 1 milliliter, 1 milliliter, nothing changes. But if you're using less than that, like 0.1, you have to factor that into the final calculation. And I think the easy way to do that is just to multiply 0.1, which is the same as 10 to the negative 1, times all of these dilutions. And that's how we get 10 to the negative 3, negative 4, negative 5. And then the positive exponent, right? So when we're counting plates, what's our range of colonies?
30 to 300.
Right. So, what's TNTC? What's TNTC?
Too numerous to count.
Right, too numerous to count. And then here Craig was so busy counting. Is that a good number, Craig?
My name's not Craig.
Oh, which one are you?
Bryant.
Huh?
Bryant.
Oh, Brian, sorry. Remember, I can't see your name, so.
Yeah.
So what's this last one here? Right, TFTC, to few to count, right? So the only really good number is this one, right? Because it's between 30 and 300. So if these are the dilution factors for the plates, let's see. Remember, we multiply, right, to get our CFUs per milliliter. So number of colonies times the dilution factor. So what's our number of colonies? Bryant. Bryant. What's the number of colonies? I guess Bryant went to the bathroom. So someone else?
Is it 3.2 times 10 to the 8?
Well, you're jumping to the end here. Right
Oh.
now, it's the number of colonies. You guys are making this hard, right? What's the dilution
32?
factor for that plate? Yeah. And the dilution factor for that plate?
10 to the 7.
10 to the 7, right? But we're not done, right? We have to put this number in proper scientific notation, which, as you said, 3.2 times 10 to the 8, right? If you make this number smaller by 1, right, move the decimal over, then we got to make the exponent bigger by 1. So you really don't need a calculator to do this, right? So questions on serial dilutions? What's that? All right, the growth curve. And so under ideal conditions, the bacteria can actually reach stationary phase in about 8 hours under ideal conditions. And then not long after that, they will enter into death. So Under ideal conditions, the bacteria can reproduce very quickly and reach stationary phase in about 8 hours approximately. And then not long after that, they'll start dying off. So this technique of the pore plate is measuring live cells, right? Because only the live cells will grow to make colonies. But when we use a spectrophotometer, and we talked about this, and measure the light absorbance, remember that dead cells absorb the same as live cells, right? And so the spectrophotometer, it does measure both live and dead cells. Now, if we were meeting for lab, we actually would have used a calculator from a website that allows you to estimate CFUs per milliliter based on the absorbance. But that was very specifically just for E. coli. And so, but that's the problem with using a spectrophotometer, is it measures the absorbance of live cells and dead cells. But the pore plate is just measuring live cells. So any questions on the plate count or serial dilutions? So next we talked about oxygen, right? Oxygen requirements. And we had melted agar deeps. We mix the bacteria throughout the agar, let the agar cool, and it traps the bacteria in place. And so we had one organism, it only grew on top. So how would you classify that?
An obligate aerobe.
Right, an obligate aerobe, right? And what was our example of an obligate aerobe?
Was it Micrococcus luteus?
Yep. And then we had two other organisms which showed the same result. Great growth on top, but also growth all throughout the agar. And so they showed us that they loved oxygen but did not need it. And so what do you call those kinds of organisms?
Those are facultated anaerobes.
Yep. And what were the two examples?
E. coli and Bacillus cereus.
Yep. Oops. Right, Bacillus cereus. And then the last one, remember that was a messed up tube? And it made CO2 gas, which cracked and broke the agar. But there was no growth at or near the surface. It was all further down in the agar. So what kind of organism is that?
That was an obligate anaerobe.
Yep, obligate anaerobe. And what was the example?
I don't know how to pronounce it, but sporgenes?
Clostridium sporogenes.
Oh, sporgenes.
Sporogenes, right? So obligate anaerobe. And then we use the anaerobic jar. Remember we had two plates. One of them was the aerobic plate, and the other one the anaerobic plate. And we had the same for bacteria, E. coli, Bacillus cereus, Micrococcus luteus, Clostridium sporogenes. And remember I said you asked yourself two questions. Where are the bacteria growing? Where are they growing better? And so what we saw So we saw that both E. coli and Bacillus cereus were growing on both plates, but growing much better with the oxygen on the aerobic plate. And that's how we knew they were facultative anaerobes. Micrococcus luteus was only growing on the aerobic plate, making it an obligate aerobe. And the Clostridium sporogenes was only growing on the anaerobic plate, so it's an obligate anaerobe. And how do we remove the oxygen from the jar? Remember, we have to remove the oxygen from the jar, and that's actually done chemically, right? Our little chemical packet, it releases hydrogen, and then a catalyst combines it with oxygen, right, to make water. Yes?
Yeah.
And then if we try to balance this, how do we balance this?
A 2 in front of the H2, and then a 2 in front of the H2O.
Yeah, so that works, right? Now, how do we verify that we created the anaerobic environment?
If you see water,
Well, but remember the agar is mostly water, and you got it in a sealed container at 37 degrees, right? So you probably see some condensation anyway. But how do we know for sure?
oh, was it if there's methylene blue?
Yep, methylene blue indicator strip, right? Remember, the methylene blue is only blue with oxygen. Without oxygen, all the blue color fades away, it all disappears. And all we see is the white paper behind it. And then we had temperature. And so remember, we used broth tubes and we incubated the bacteria at 10 degrees, 20, 37, and 60 degrees Celsius. And we observed two characteristics, either turbidity and sediment, right? And so what if you had an organism that grows at 20 and 37, especially good at 37?
It's a mesophile.
A mesophile, right? What if the organism only grows at 60?
Then it's a thermophile.
Yep. And then we had one, it really, really liked 20, but it grew a little bit at 10.
That was a psychrotroph.
Yeah, remember psychro, not psycho. Right? It really does prefer room temperature, but it tolerates the cold. A 37 is way too hot for it, right? And so what was the example of a mesophile?
E. coli.
E. coli. Our thermophile was Bacillus staurothhermophilus. And then what was left was the Pseudomonas fluorescens as our psychotroph. And remember, no spelling, it's all multiple choice, right? And then let's talk about growth patterns next, and we'll finish with transformation. So on the growth patterns, remember for agar cultures, we had consistency and pigment. What were the two consistencies?
Mucoid and dry.
Right, mucoid and dry, and most bacteria tend to be which one?
Mucoid.
Mucoid, right? So some bacteria are very, very mucoid, others not so much, but we still call them mucoid. And then if they look rough, crumbly, we say they're dry or friable. And for pigment, do all bacteria produce a definite pigment? Do all bacteria produce a definite pigment?
No.
No, right? But some do, right? And they can be very colorful. And if they produce a colorful pigment and it stays inside the bacterial cells, we call that intracellular. If a pigment is secreted to the outside, we call it extracellular. And what's the purpose of these pigments? Is it to make bacteria pretty?
No, it could be for a lot of different reasons. Like
Such as?
to protect them, or they're antioxidants, or
Right, yes. So,
make them pathogens.
protection is a good umbrella term. It can protect them from UV. It can protect them from oxidative stress, free radicals. It can protect them from predators. And so the pigments do serve a purpose, right? They're not just to make the bacteria pretty, right? Bacteria are not vain like human beings, right? And for broth, we had 4 characteristics of broth: a pellicle, a turbidity, sediment, and flocculent, right? So, pellicle was a layer of growth across the surface. Of course, sediment was on the bottom. A turbidity means cloudy, and flocculent was little clumps, right? So just be able to recognize those characteristics when you see them. And then there was transformation. So remember we had a plasmid, right, the pea glow, and it had three genes of interest. And a special promoter, right? What was the blah gene? What is that code for?
Is it for destroying antibiotics?
Right, so the enzyme is beta lactamase, right, which destroys the penicillins like ampicillin. And this selects for the transformed bacteria, right, the transformants. And that's how we know transformation occurred, right, because the bacteria are now resistant to beta lactamase. Yeah, that's a dash and an L. So beta lactamase. Of course, GFP is the green fluorescent protein. ARAC is another protein. And then PBAD, a special promoter, right, for the green fluorescent protein gene. Is PBAD normally on or off? On or off?
Off.
Off, right? It needs 2 things to get turned on. What are those 2 things? Needs two things to get turned on. What are those two things? You got to tell me. So what are they? It's going to be a lot of dead air. So what two things are required to turn on PBAD?
Was it the arabinose?
Right, arabinose and the ARAC protein. Where does the ARAC protein come from? Where does ARAC come from? It comes from the bacteria because now they're making it, right? They have that gene. And the arabinose has to be given to them in the agar. And so you have all right, so this is the ARAC protein. Arabinose binds to it, and then together they both bind to PBAD. This turns it on, and so now RNA polymerase can hop on and start transcribing GFP. Right, so you need two things to turn on PBAD. The ARAC protein, which the bacteria are making, and the arabinose sugar, which has to be provided to them. And the arabinose binds to ARAC protein, and together they bind to PBAD. That turns it on. Now RNA polymerase can hop on and start transcribing the green fluorescent protein. So remember, we had 4 plates, right? 2 plates, no plasmid, and the other 2 plates had plasmid. This was just LB, ampicillin. This last one was LB, ampicillin, and arabinose. Right, so we had lots of growth on the LB plate. We had no growth on this plate, right? What does this plate tell us?
That ampicillin kills the bacteria.
Right, that ampicillin kills the bacteria. So in other words, E. coli are sensitive or susceptible to the ampicillin. And this plate here just tells us our E. coli are alive and they grow on LB agar. And so what do we call these 2 plates in this experiment?
The control.
Right, they're controls. And these 2 plates are the experimentals. And of course we had, right, growth all over. Remember, these were green. So we have bacteria growing in the presence of ampicillin. And so that means that these bacteria are now ampicillin resistant. This plate here tells us transformation occurred. This plate here shows us gene regulation, right? Because the bacteria are also growing in the presence of ampicillin, but we have fluorescent green colonies, which means the green fluorescent protein gene has been turned on. It's turned on because we have arabinose in the agar, and the bacteria are making the ARAC protein. On this plate here, we only have the ARAC protein that the bacteria are making. There's no arabinose in this plate, so no green fluorescent protein. There's only one thing is available here, but both things are available in this plate, right? So GFP gets turned on. And so what do we call it when we have from ice to heat to ice? Along with calcium chloride, what do we call this? Remember the heat shock? And so that allows the membrane to become permeable. It makes the cells competent, right? And so they can absorb the plasmid. And so,
I don't know if it's just me, but I can't hear.
Oh, you can't hear me well enough? Is this better? Is this better? Can you hear me now? I guess not. Yeah, okay. Yeah, sometimes that happens where, the sound gets lost. So hopefully Rachel will be able to come back and check her microphone and speakers. So then, after the heat shock, we gave them some food and 10 minutes before we spread them on the surface of the agar plate. Why did we do that? Right, because after the heat shock, we cooled down the bacteria, right? The plasmid had already gone inside, so they were transformed. So why then did we give them food and time before we spread them on the agar plates? So why did we do that?
Was it to give them a lag time? Lag phase?
Well, they were in lag phase, right? So we were giving them time to sense the environment, see what's there, turn on genes, especially to transcribe and translate the beta-lactamase gene, right? So we had to give them time to actually start making beta-lactamase. Otherwise, if we immediately put them on plates containing ampicillin, they would have died, right? So we had to give them time and food so they could make some beta-lactamase. So does that make sense to everyone? Rachel, did you make it back?
Yeah, I'm here. I didn't know if it was just me. That's why I said something.
Yeah, it was just you. But,
I guess my computer is acting up today.
oh, that happens sometimes. So now remember on the 2+ DNA plates, we had the bigger colonies. And then we had some little colonies growing around them. What were those and why were they there?
Were those the satellite colonies?
Right, the satellite colonies. And so what were they exactly?
Because the main bacteria destroyed the.
Well, but what were the satellites? I mean, what are they? Tell me what they are.
They're bacteria,
Right, they're just plain E. coli, right?
right?
And so we already showed that plain E. coli is killed right by ampicillin. So then how come they're growing on plates with ampicillin?
Because the bacteria that were transformed destroyed the antibiotic and that around it and it allows for the other colonies to grow.
Right. So the transformed colonies are making so much beta-lactamase that they're destroying the ampicillin in the agar immediately surrounding those colonies, and that creates a safe zone. And so non-transformed E. coli are able to survive and grow, not well, but as long as they stay right next to the transformed E. coli, they can survive. So does that make sense?
Yeah.
So any other questions about the lab exam?
How many questions are there?
There's 15 multiple choice questions and one bonus.
Okay, thank you.
So anything else?
So we can submit the unknown as a PDF.
Yep.
Okay.
Remember that's due by tomorrow night. Okay, so Wednesday we'll have some lecture review for the exam. Any questions you might have about anything. And then at 1 o'clock we'll start the lab exam. So remember you have to sign on to collaborate and turn your camera on. And then I can finally start learning who you guys are by seeing what you look like. And then you start the exam. So anything else? Well, that'll do it. If you have no questions.
You,
All
Professor.
right. Okay, Bryant. Okay, I'll see you guys Wednesday.
Thank
Bye,
you.
Christiana. Bye-bye, Rachel.
Bye.
Please. What do you mean keep your camera on? I mean, when you finish taking the lab exam, you're done, so you can turn it off.
Are you just using the cameras to make sure that we aren't using papers? Because like if we use our phone, won't we be looking somewhere else?
Well, the only reason why you would use your phone is maybe to use a calculator, right?
Oh no, I'm probably gonna use a calculator, like a actual calculator.
Oh, natural calculator. But then you don't need to use your phone, right?
No, because the camera on my computer is weird.
It's weird.
Yeah, like it's not clear at all.
We'll turn it on right now. Let me see.
No, thank you.
Just turn it on.
No, I look crazy.
It's all right. All right, well, make yourself look nice for the exam. All right. And then make sure you clean the camera lens. You know, most people never clean any lenses on anything like their smartphones, but make sure you clean the camera lens because it could be, you know, dirty. All right. All right, Sarah. I know Christina would love to turn on her camera, right, Christina? I know what she looks like. All right, Craig. I'll see the rest of you Wednesday.